Hi, everyone, and thanks, Irene, for agreeing to give a Build a Cell seminar, and you can get started.
All right. Thank you, Kate, for the invitation and putting together such a vibrant community. Um, this is kind of fun for me because it's kind of the end of a chapter on protocells for me. I feel like my lab has started for multiple reasons, kind of like migrating toward working on phages, and it was nice to go through some of this work and try to put it together in some themes. So. What I'll tell you guys about is what we've been doing on fitness landscapes for RNA and the ways that membranes can help RNAs in the RNA world. I'm sure that a lot of you have already seen this type of diagram and are aware that there's a subset of people in the synthetic cell community who are interested in protocells or synthetic cells from the perspective of the origin of life. So this is a mural from NASA where I think it's in the Ames Research Center, and they're trying to capture all the important events, transitions that together formed the origin of life, going from like kind of molecules and prebiotic chemistry to an RNA world to the simplest cells, and on to the pinnacle of humanity, which is astronomers, apparently. So what I'll talk about is kind of this RNA world area of the diagram. And again, I know a lot of you are aware of this, so I'll just briefly introduce this idea relatively quickly. This is the idea that RNA has this kind of dual capability of acting as enzymes or biocatalysts, and also encoding information. And this is a nice, I think, demonstration of how powerful that is. You know, it's as if the words that you use to describe something themselves would fold into that thing. And so this is that kind of dual capability of RNA. And this idea is really old, the idea of this RNA world, which would kind of dispense with the need for the kind of DNA and RNA, DNA and protein parts of the central dogma, goes back to Francis Crick, Leslie Orgel, and Carl Woese, who proposed after noticing the complex structure of tRNA, of transfer RNA, before actually knowing anything about catalytic properties of RNA, proposed that RNA could be biocatalysts based on the complexity of the structures. And then there's been a variety of pieces of kind of circumstantial evidence that people point to as supporting the idea of an RNA world. There's how RNA and bits of RNA, especially adenine and ATP, kind of pop up in metabolism, how there are still catalytic RNAs, ribozymes, and how the ribosome itself, which is protein and RNA, but the catalytic center is really just RNA. So this machine for translation being an RNA making proteins is kind of called the smoking gun by Tom Cech. But even if you kind of don't care about the origin of life, I think it's still a nice model system for synthetic cells because of all of these same properties that make it interesting for origin of life, and being the first cells also make it interesting for synthetic cells. And the understanding of RNA has kind of evolved over time. At first, RNA just seemed like the messenger. And then in the 80s, with discovery of catalytic RNAs, it does some things by itself also. And then, microRNAs, siRNAs, RNAi, so many things going on. It's doing a lot. And I don't know, maybe now it seems like really we're still living in this RNA world. So we were interested in evolution and understanding, like trying to systematically understand evolution of RNA. So first, let me just mention the concept of a fitness landscape. Again, I know this is review for a lot of people, but I'll just mention it. So if we imagine that we have sequences, sequence space, this is like n-dimensional space of all possible sequences. And if we have fitness values for all of those sequences, then we have what's called the fitness landscape. So on this fitness landscape, you have these kind of high areas, peaks, which correspond to like highly active or highly fit sequences. And then you have these lower areas that are these inactive sequences. So natural selection on the fitness landscape is this process of exploring through mutations, local area, local sequence space, or maybe more distant through recombination, et cetera, but exploring sequence space based on the current population, and then moving the population up toward higher fitnesses and hopefully discovering these peaks. So we wanted to understand what these fitness landscapes look like for RNA, since at the very beginning, presumably there was kind of a random exploration or almost random exploration of sequence space, and some of these fit sequences had to be discovered. So we wanted to map the potential pathways for evolution, and RNA was a great model system because even though for genomes or organisms or the synthetic cells that a lot of other people work with, it's the sequence space is much too big to really examine in the laboratory. It is possible to get like 4 to the 21 possible sequence. It is possible to kind of cover that sequence space in a laboratory experiment. So it was a nice system for looking at fitness landscapes of molecules from that perspective, because the sequence space itself is pretty small compared to proteins or other interesting biomolecules or even organisms. So one of the questions we wanted to answer by looking at these fitness landscapes was how well can natural selection optimize fitness? Like how rugged are these fitness landscapes, and are they connected to each other? If you maybe found a local optimum, could you get to the global optimum? That kind of thing were the questions we're interested in. So to map these landscapes, again, it's tractable for relatively small RNA sequences. We would take a random library, and this is looking very much like CELEX. We would start out with a random library of RNA sequences and then pass it through some kind of selection, either like an aptamer type selection or a ribozyme type selection. I'll show you one of our selections. And the idea is that we decrease the frequency of these inactive sequences, we keep those active sequences, and ultimately the goal is to kind of use this data to discover the shape of this fitness landscape. So the activity that we decided to look at was based on aminoacylation of RNA. So as you know, protein translation has many pieces to it, but one of those pieces is preparing the charged tRNAs. So attaching an amino acid to an RNA is an important kind of prerequisite for protein translation. So this was an interesting activity from the kind of importance of it in biochemistry. But then also what was really nice about this activity is that a few different groups had already shown that RNA was able to self-aminoacylate or to catalyze this reaction. So that gave us like a high confidence that a selection based on this activity would work. So we're looking for self-aminoacylating ribozymes. And to get this started, we worked with Ziwei Liu and Robert Pascal at the University of Montpellier, where they had proposed these activated amino acids. So these are 5,4-H oxazolones, and you can see the kind of amino acid backbone here. This is going to be an amine, and we'll have a carboxylate here. And our side chain will go here at the R1. And then at R3, Ziwei could put in something convenient for us, which was a biotin, which will be a nice handle for our selection. So they proposed these activated amino acids based on some considerations from prebiotic chemistry and like what molecules were present in volcanic gases and that kind of thing. So it was nice from that perspective, but also for us in the lab, it was nice because it was stable, had, you know, a lifetime that was significantly longer than what than like adenolates in solution, other adenolates in solution. So we liked these oxazolines for their kind of practical properties as well. Ziwei had also shown that these oxazolines react slowly with nucleotides. So here is a tyrosine analog. Here you can see the side chain has been methylated, kind of protected on the OH. It is our tyrosine analog, and it reacts. Ziwei had shown already that it reacts slowly with mononucleotides. So it's a thermodynamically downhill reaction. We just needed to find an RNA sequence that would catalyze this reaction. So my student Abe made a random RNA pool from a random DNA pool, and this is an in vitro selection setup. So we had an RNA pool covering sequence space. So we think it has 4 to the 21 different molecules in there in at least 100-fold coverage. And reacted it with our oxazolone that Ziwei made. So here we've got a biotin, and that's something that we can hang on to later. And then we've got our side chain here. So any RNAs that react with the oxazolone will get biotinylated, and then we can pick out those biotinylated RNAs with streptavidin beads. And then our selection can amplify those, and we can go through multiple rounds there. So just to give you a little background on how we get the catalytic activities out of the sequencing data, we worked on a technique that we called kinetic sequencing, which was the idea like you've got a kind of medium complexity pool, so maybe some thousands or tens of thousands of sequences in there. And we wanted to assay all of those sequences for their catalytic activity, their KCAT. And to do that, we took the whole pool, aliquoted it into different reaction conditions, and then conducted the reaction. So this could have been like time points. I think that's probably the easiest one to think about. But in our case, because of degradation of the substrate, we use substrate concentration. So we had like different concentrations of substrate. And then you can fit a kinetic curve to these sequences that you collect, the biotinylated sequences that you collect from each of these reacted pools. So this was a high throughput kind of assay for catalytic activity. And so we would get this number of activity associated with each sequence that was present in this kind of medium complexity pool. So that's reasonably correlated with the catalytic activity we would measure by more like traditional methods, by gel shift assay. So by K-seq and by gel shift assay, if we compare the catalyzed rate to the uncatalyzed rate, those two measurements lined up pretty well. Okay, so back to one of our original questions. We were wondering about the connectivity of the fitness landscape. So I like to kind of go back to this. It's simplistic, but it kind of captures the idea view of early evolutionary biologists, John Maynard Smith. And he was saying that if a natural selection is going to occur, in particular for this optimization process, then there needs to be a continuous network that can be traversed by unit mutational steps. So like mutations leading from point A to point B without going through a really low fitness intermediate. So the idea is that we want to find these pathways that are viable so that our organism doesn't lose fitness or our molecule doesn't lose fitness while going from point A to point B. And in the 2000s, there had been some computational work which really inspired us on this question for RNAs, and they were looking at RNA secondary structure as being kind of the fitness of the molecule. So they asked, can we get from arbitrary point A in sequence space to point B in sequence space, both A and B fold into the same secondary structure? Can we get from point A to point B, making single mutations and preserving that secondary structure the entire way? And what they found was pretty remarkable to me, at least. They found that the answer is usually yes, you can preserve the secondary structure traveling from point A to point B along the entire way. So this kind of gave the idea of a evolutionary network that permeated sequence space and allowed like a high degree of connectivity throughout sequence space, going from one optimum to another optimum, kind of not losing activity or not losing secondary structure. So the question is, for an experimental setup where we're measuring the activities, the catalytic activities for this amino acylation reaction, which we're using as our model, for that, do we still see this kind of continuous network? So here's an example of one of the better kind of pathways that we were discovering. Here's these colored dots indicate some of the higher activity sequences that we found, and the intermediate points, these black dots, this is just one example of a pathway, a relatively good pathway, I guess, going between these different local optima. And what you can see is that some optima seem like fairly well connected, not losing too much activity here from 1B to 1A, and here are the sequences on the x-axis. But going from, let's say, 1A to 2.1, there is this very large jump where you'd have to get from here to here without kind of passing through this valley, and that would be very difficult to do. So what this gave us, and this is not just this particular pathway that I'm showing here, but this was kind of general across the landscape, looking for the best pathways. They always had these kind of valleys in them. So here's a representation of like many pathways, like several pathways between these optima, which are the big circles here. And I guess what is easiest to see is that if you focus in on the global optimum of the landscape, that's kind of separated from the rest of the landscape by these red dots. And these red dots are indicating places where there's very little catalytic activity. And then these dotted lines mean that many mutations are required to get from one dot to the next. So there's this large valley between 2.1 and 2.2, this kind of optimum, and the rest of the landscape. So we thought that this kind of indicated a kind of a property of frustration for evolution for these small RNAs. And just to kind of connect it a little bit to places where you might have seen frustration in other contexts, there's also people talk about frustration in protein folding, where there might be, in the best case scenario, you have all their different conformations leading to the lowest energy state, but maybe in reality, you have a situation where there are many local optima to get trapped in. And then there's the classic model for frustration in an antiferromagnet, where in this material you're trying to optimize the nearest neighbor interactions by having up next to down. And that's totally possible if you have like this sort of geometry. But if you have a geometry like with triangles, then in this case, you can't find any, you don't know how to assign the ups and downs here. So you have many different solutions, I guess, that all are suboptimal, or they all have similar energies. So what configuration you actually have in a particular spot is highly contingent on whatever factors set this initial configuration. And that's just like in a fitness landscape, maybe the outcome that you ultimately get, what we see is that it would depend a lot on exactly where you started on this landscape. So where on this landscape did you start with your RNA world would be very influential on what outcome you ultimately got. You wouldn't necessarily expect the global optimum to emerge. Okay, and this is just a fun XKCD illustrated social frustration, I guess, where there's this social network and they all have to sit down in a movie theater in a line. And they're trying to, optimize too many constraints at the same time. So you end up with situations, there's no optimal configuration, and here's a third wheel kind of getting stuck in the middle. Okay, so that is just kind of the intro to fitness landscapes that we looked at. And then once we had these techniques, we sort of thought, well, what else can we do? And one of the things that we could do is to change up the substrate. So Ziwei, together with Evan, my student, made a bunch of different substrates, different side chains. So you can see like tryptophan, leucine, valine, etc. And here we kind of just applied our cudgel of kinetic sequencing to all of these different substrates, and so measured the activity of our ribozymes for all these different substrates. And here you can just see by eye even that some ribozymes have more promiscuity, like 2.1A is very promiscuous, whereas like 1A.1 has a kind of specificity, seems to prefer the aromatic amino acids. So on a larger scale, with kinetic sequencing, we have the catalytic ratios for each substrate and each point here is one ribozyme sequence, and these are two substrates just kind of to illustrate the correlation in some cases and then lack of correlation in other cases. And so we have this for all the different comparisons that you could make. And this also illustrates like how some of these families are more promiscuous than others. So we have like low activity in the kind of family 1 zone, but higher activity in the family 2 zone across different possible substrates. Okay, so what do we want to use this data for? There is always this question for different proteins, and like different systems might have different answers to this question, but we thought it would be interesting to know for ribozymes whether activity and specificity are represent a trade-off. Like there's kind of this idea that if you evolve something to be more active, then it also becomes more promiscuous. And the reason for this intuition is that in like kind of translating this intuition into our case for ribozymes or self-amino isolated ribozymes, if you add contacts to the substrate to improve specificity for the correct substrate, so this maybe mutant ribozyme has this more stabilized ribozyme substrate structure, then that increases the activation barrier. So that decreases the rate. So this would give you a trade-off between increasing specificity and decreasing rate. And that's kind of like, that feels intuitive because it says like there's no free lunch, you can't get greater specificity without giving up some activity. So the prediction here is that the higher activity sequences ought to be more promiscuous or less specificity. So what do we see in our data? This is an index of promiscuity, basically like a Shannon entropy type of measurement against the total overall activity of each ribozyme. And what we saw in general was that the higher the activity is, the less promiscuous it is. So higher activity to the right and less promiscuous going down, with promiscuity index going down, which is the opposite of that kind of like, initial intuition. So digging around a little bit, it turns out that the other way to think about gaining specificity, in sort of two extremes, you could either increase contacts in the ground state, or you could increase contacts in the transition state. And if you increase contacts in the transition state, then you increase specificity, but you also lower the activation barrier. So this would give you more specificity and a faster rate. And this would give you the correlation of higher activity also being more specific or less promiscuous. So that's a possible explanation for what might be going on with our ribozymes, kind of following closer to this paradigm than to the previous paradigm. And regardless of what the actual mechanism is, the fact that there is this correlation says that if you select for higher activity, then you should also get higher specificity for free, basically, because if we look here, you're kind of pulling the population over here, and that will become less promiscuous because of the this correlation. Okay. Now, that was all a little bit prelude, sorry, for synthetic cells. So let me talk more about synthetic cells now, which is what is a little closer to what you guys are interested in. So cells, we know, are very important for life, so let me not kind of belabor that. But I also just want to emphasize that the sort of biophysical environment inside a cell or a vesicle is different from the environment in bulk solution. And we know that from a lot of, biophysics work that's gone on in the past few decades. So the model for protocells that we're using, I know Kate is probably intimately familiar with this, and others are too. We're using fatty acids primarily, being inspired by the content of meteorites. And then we also use phospholipids sometimes as a nice model system for our lipids. And we're interested in understanding what happens inside to encapsulated RNAs. So the first experiments we ever did on this were to encapsulate the malachite green aptamer. And this is an aptamer that binds to the small molecule malachite green and causes the molecule to fluoresce when it's bound to the aptamer. So activity is really easy to monitor by fluorescence. So we simply just asked if we have malachite green aptamer outside the vesicles versus inside the vesicles, does encapsulation affect binding? And the answer is yes. There is a significant, although not incredibly large, but it's still a significant shift in the binding curve for the malachite green aptamer. So outside the vesicle, you get this binding curve in gray, and then it shifts to the left in blue. And that indicates a greater activity of the aptamer inside the vesicles. And then if we look at a melting temperature, or really a transition temperature because since we're following fluorescence, if we look at the kind of local melting, I guess, of the pocket, then you can also see that outside vesicles, you have a particular melting temperature, but inside vesicles, this is shifted over, indicating greater stabilization of the aptamer when it's inside vesicles. And then we also looked at conformations using shape, and we also looked at crowded conditions. But what we were looking at, what we were seeing is that as you encapsulate the aptamers, you get a shift of the conformation toward the more folded state. And this was also borne out in a couple of other types of structures. So here we were looking at just a hairpin. I thought, okay, this is about the simplest situation we can think of is a stem loop structure. Can we measure any change in the folding in the melting temperature, which we're monitoring by fret between the two ends here? Yes, if you're inside vesicles, which is in red here, versus outside vesicles in blue, you see this shift from in several degrees Celsius. You see a shift in the melting temperature increasing, indicating greater stability of this stem loop structure inside vesicles. Then we looked at this pH switch, which was already reported in the literature as triplex formation, pH sensitive triplex formation, which can be monitored by quenching and unquenching of a dye attached to the 3' end. And we looked at the pH that was required to cause unfolding of this triplex. And it turns out that you have to add more base, significantly more base, if the switch is encapsulated compared to if it's outside vesicles. So you have to put greater driving force in order to unfold the switch, this triplex, when it's inside vesicles. So that's the red curve compared to the blue curve. Okay, and how about a ribozyme? So we did this in collaboration with Sabine Muller, and she's an expert on the hairpin ribozyme and knew exactly what constructs to use here. So here, the hairpin ribozyme is a self-cleaving or also ligating ribozyme, and it can be labeled in such a way that a important intermediate, the docked intermediate of this ribozyme, brings together the two fret dyes, so we can monitor docking. This is a obligatory intermediate on the pathway on the catalytic pathway. So this docking interaction can be monitored by fret. And what we see is that the docking itself is increased by the fact that the ribozyme is encapsulated. So outside again in blue and encapsulated in red, showing that you have more docking and faster docking when the ribozyme is encapsulated. That also translates into greater activity since the docked intermediate, the formation is important for catalysis. So here we have a mutant C25U of the hairpin ribozyme that normally is docking deficient. So it's a folding deficient mutant that normally shows this blue line being unable to dock. But if you encapsulate this mutant, then you can kind of rescue some activity. You can see there's some docking because the mutant has been encapsulated. And this translates into some ligation that's happening inside vesicles, but not outside vesicles. Okay, so just to give some intuition into what is going on here, we think that this is a type of confinement or excluded volume effect. And the idea here, if you haven't seen it before, is that normally, let's say you've got a nicely folded RNA in the folded conformation. It can exist in many configurations. And some of those configurations, if you encapsulate it now, if you kind of impose this boundary, some of these configurations are no longer accessible. If you have an unfolded kind of not compact, kind of flopping all over the place RNA, and then you apply the same boundary constraint, then only a few of the configurations are still allowed. A greater fraction of these possible configurations are disallowed in the when you have the kind of unfolded versus the native state. So in both cases, I guess you could say that there's some destabilization if you're going from kind of free solution to being confined. But if you're comparing confined for the folded versus the unfolded, then there's a relative stabilization of the folded versus the unfolded because you have this kind of greater exclusion of possible states in the for the unfolded version. Hope that was a decent explanation. Okay, so the overall effect is that we are looking at kind of a free energy diagram for the unencapsulated versus encapsulated. And if you have some particular free energy diagram for unfolded versus folded and a, RNA then gets encapsulated, then there's going to be this relative destabilization of the unfolded state, which will give us relative stabilization of the folded state. So we have better folding, which might translate into better activity, at least better compactness, which might translate into better folding and better activity. And we've seen this in several different situations. Okay, so I think this is the last thing I'll talk about is whether encapsulation affects RNA evolution. So we've already kind of seen some things where like you can rescue a mutant sequence by encapsulating it. And we wanted to understand for an entire population being evolved, does being encapsulated affect its evolution? Just to refresh your memory, we've got all of these different self-amino acylating ribozymes, and we can evolve them by an in vitro selection procedure using these biotinylated oxazoles. So the first thing that my postdoc Chen did was to encapsulate these a few self-amino acylating ribozymes, and then add BYO, the substrate, and then ask what's the activity when the ribozyme is encapsulated versus when it's outside the vesicles. So inside vesicles in blue here, higher activity versus in red here, which is outside the vesicles, lower activity. So we can see this difference, and that's what we expect based on all the things I've already shown you. So that's not too surprising. But now we want to do it on the entire kind of population, kind of surveying whether we have this general trend of increased activity, and or whether we might see any exceptions from that. Okay, so right now we're looking at 5 families of ribozymes, and the activity inside vesicles is on the y-axis, and the activity outside vesicles is on the x-axis. So the first thing that you notice is that there's a really good correlation between the activity inside the vesicle and outside the vesicle. And that makes a lot of sense. The fact that the RNA is encapsulated is kind of affecting the RNAs, all the RNAs in a similar way. But one thing that we kind of zoomed in on was, let's just look at this red plot for a second. For this particular ribozyme family. Yes, there's a really good correlation, but if we look at the x equals y line, that's this dotted line. And if you trust our measurements, then you would say, okay, let's fit the red dots. And actually, if you fit the red dots, the slope of this fit is greater than 1. So it's not really the x equals y line. It is deviated where these higher activity sequences have a greater increase in activity when they're encapsulated compared to these lower activity sequences. Okay, so our slope of fit is a little bit greater than one, and we see that across these different ribozymes. The slope for fitting is greater than 1, and what this means again is that the high activity sequences are getting more benefit from being encapsulated compared to these lower activity sequences. So these high activity sequences kind of peeling away from this kind of general population of these lower activity sequences, they're getting a greater benefit. And it's, I would say it's small, but it's consistent. And a small but consistent effect in evolution still gives you a pretty big effect. What our prediction here was that there's greater variance among the sequence activities when they're encapsulated. And this is seen by how you have this higher activity population kind of peeling away from the x equals y line. So greater variation, greater variance in the activity. And there is a theorem in natural selection, Fisher's fundamental theorem of natural selection, which says that the rate of adaptation that's due to natural selection is proportional to the genetic variance. And this theorem is like very infrequently used because it is so simplistic. There's no environmental variation here. There's nothing here except for a natural selection and genetic variance. And this is okay for our in vitro selection experiment. So unlike any lifelike situation in our in vitro selection experiment, this probably captures most of it. So we expect that the rate of adaptation is going to be proportional to the genetic variance. In other words, if you had a lot of variation, then your population converges quickly. Whereas if you had like very little fitness variation among sequences, then it would take a long time, many cycles, a lot of cycles of enrichment to get the population to converge. So we're expecting the overall effect to be that if you have your RNAs encapsulated, then you should get faster adaptation because of this increase in genetic variance. Okay, so what do we see if we encapsulate the RNAs and then do the in vitro selection experiment with encapsulated RNAs? What we're looking at here is a diversity measurement, diversity index, and that's the probability that two randomly selected sequences are different from each other. So that's really high in the beginning because we have a large library of different sequences, and then it decreases as the library converges. So let's look first at the non-encapsulated situation. So we see that there's a particular rate of convergence. And then looking at the encapsulated version of the same selection, we see that convergence happens a couple of rounds earlier. And it's a short selection, just 5 rounds. So going from, saving 2 rounds, I guess, is a pretty substantial change in the rate of adaptation. Just to sum up these different projects that I've talked about, the overall idea is that the vesicles are kind of chaperoning the RNA, very primitive kind of chaperone that might have been available as a mechanism for early life, kind of favoring through the excluded volume effect, confinement effect, favoring the folded conformations and giving you relative stabilization, maybe of some poorly folded RNAs. And then that kind of less maybe intuitive finding is that because there's this greater variance among activities or fitnesses when you have the RNAs encapsulated, then you get a greater rate of adaptation. So maybe in addition to having kind of some absolute benefit to encapsulating your RNAs, you might also get a benefit of having your RNAs converge faster onto the fittest genotypes. Okay, so I want to thank all the folks who contributed to this. At this point, they've all left the lab, gone on to better things. But what I've shown you is the work of several folks who contributed to protocells, fitness landscapes, and all those thoughts. I also want to thank our collaborators, Ziwei and Robert, who were our prebiotic chemist experts, and Uli Muller and Jerry Joyce helped us develop K-seq, and Sabine was our hairpin ribozyme expert. And I'll stop there and happy to discuss or take questions.
Thank you. Thank you so much. That was really awesome. There is a lot of questions in chat. Can you see the chat questions?
Oh, yeah. Let me see here. Maybe if I stop this here. Oh,
Read
okay.
the question out loud before answering because people watching the recording don't see the chat.
Okay. What are the limits of probing fitness landscape and artificial selection experiments like you're describing? Can you only probe around existing function, or could you discover novel mechanisms that nature never discovered? So the selection, I guess, is only focusing on phenotype. It's not trying to get a particular mechanism. So I would say that like the selection experiment itself is kind of agnostic about what the mechanism is. And I think a lot of people have seen in their in vitro evolution experiments completely novel things coming up. Like Burkhardt-Selig has a great example of a ribozyme that catalyzes the Diels-Alder reaction. And that was discovered, long before a protein to do the same reaction was developed. So I think absolutely in general, to answer that question, novel mechanisms can emerge during in vitro evolution because there's no bias toward existing like genomic sequences. Okay, let me go on. You observed higher activity inside liposomes. Do you think this is mostly due to crowding, or is there other special biophysical effect from vesicles? Yeah, I think it's an excluded volume effect. Yeah, maybe I guess you anticipated that answer before I tried to explain the mechanism. So thank you. Dieter, if I understand, the effect shown rather works for small vesicles, 2 to 10 RNA molecules. Not expect the effect for micron size. Yeah, definitely if this effect should be greater for greater confinement. The micron-sized vesicles, if it's a sphere, yeah, I am not sure you would get much confinement from the membrane part of the confinement. Maybe you would crowd it with other things or whatever, but yeah, I agree that if it's a micron-sized vesicle, we would not see much effect. We did try some larger vesicles, like we did extrusion to maybe 400 nanometer vesicles, I think, and we do see a decrease in the size of the effect. So yeah, maybe you could kind of like make pancake shaped micron sized vesicles and try to get some effect there. But you're right. I think for a sphere, micron size probably insignificant effect. Um, is there any effect for vesicle composition? Yeah, we looked at this. I didn't talk about it, but we were definitely curious about this question also. Is there any effect from vesicle composition? So if you focus in on just the what confinement the vesicle is providing. So if you have the control of RNAs outside vesicles and RNA inside vesicles. So those could be POPC vesicles, those could be fatty acid vesicles, whatever. Then we've found for all the compositions we've tested that relative stabilization is the same. But if you just look at the effect of the lipid itself, so say you have like the lipid in a buffer, salt buffer solution, or I'm sorry, you have the RNA in a salt buffer solution and the RNA in the presence of the lipid, like outside the vesicles, just in the presence of that lipid, then the lipid itself can have different effects on the RNA. So I think an extreme example of that would be like a cationic lipid. You probably get some association or complexation or aggregation. But even with like negatively charged lipids, you can get some like denaturing effect, what would appear to be like a denaturing effect on the RNA from just the presence of the lipid. So that's a kind of long way of saying, and you also get that kind of thing with crowding agents. If you probably several people in this community have tried to do crowding agents to improve activities, and you can get different effects depending on what crowder you use because of chemical interactions with the crowding agent. I think it's a similar situation. So in all the experiments I showed you, we were always comparing inside vesicles versus outside vesicles to control for that chemical interaction effect. Okay, let me see. The next question, self-amino-acylating RNA inside and outside vesicles. Since there is a membrane, wouldn't the RNA interact with a membrane from outside? Yes, yeah, so that's actually very related to that previous question. Didn't I look at encapsulation for other catalysts like peptide enzymes? Yeah, I think that people have done all kinds of things with proteins, like nanocapsules and that kind of thing. So I think that there's a nice literature on that. I don't know if anybody has encapsulated proteins in the kind of vesicles that we were working with, like with protocell type membranes. There, I'm not sure. I bet you, like with everything, things are more complicated with proteins because you have the hydrophobic, amino acids and side chains and everything. But I think there's a literature at least on encapsulation in like more inorganic substrates. I would bet that there are experts on encapsulating proteins, and I'm just not very familiar with that literature. What are the limits of probing? Oh, wait, I think oh, thanks, Kate, for adding that. Kate put uh, a link in the chat. John, okay. Last question here is John Maynard Smith's theory of continuous evolution. Isocyanates, functional. Yeah, so that was totally okay. So the question is about like whether you had like a duplicated gene or two like very similar genes, and one of them can kind of keep doing the housekeeping function, and the other one can go off and evolve in some different direction without hurting the fitness of the organism. Yeah, I think that's absolutely right, and that's very important for organism level evolution. What I was talking about was just that very simplistic view of like we've got like one collection of RNAs that are all kind of around this particular peak, and would we be able to without say gene duplication or recombination, which are not considered in that like quote that I put in there, would we be able to connect the optima? But you're absolutely right. If you had gene duplication and you made your genome longer, this is that's absolutely a way around these valleys. Okay, well, I think I've gone through all the questions and I really appreciate your attention. Thank you.
Thank you so much. And I particularly appreciate the protein and liposome composition speculations because I feel like that's this is the directions people are going to look into, inspired by your RNA results. Okay.
Cool. Cool. Yeah, thank you.
All right. Thanks, everyone. Thanks for a great discussion, and thanks, Irene, again for a wonderful talk. And see you all next week. Bye.
Bye.
A good one. Bye-bye.
Bye.